Welcome to the first of two lab videos related to Lab 5. Today will be Lab 5A as we begin an introduction to the muscular system. Muscles. What do they do? Well, you know that they are attached to your bones, and by contracting, by changing shape, that they move your skeleton. You are aware that as your heart beats and as your muscles move throughout your digestive system, for example, you are moving substances. You're helping to move blood, hormones, nutrients, water throughout your body. And then finally, when you are cold, you shiver, creating heat, and the circulatory system helps to distribute that heat. What are the general characteristics of muscle tissue? You learned the general characteristics of the epithelial tissues and of connective tissues in a previous lab. Remember, epithelial tissues were composed of tightly packed cells with no blood flow, avascular tissues, whereas connective tissues were loosely arranged cells surrounded by a ground substance in which there were protein fibers. Likewise, there are four characteristics of muscle tissue that make it unique. Number one, it's excitable. That is, it is responding to signals from the nervous system. When muscle gets excited, when it receives that electrical signal from the nervous system, it contracts, it shortens. And as it shortens, it pulls bones or squeezes to contract structures. Muscle is elastic. That is, muscle, once it contracts, can return to its original shape. It's elastic. It's like a rubber band in a way. And finally, muscle tissue is extensible. As we look at muscle groups over the next few weeks, you will see that many muscle groups are opposing each other. So, for example, when you contract the biceps brachii, the triceps brachii simultaneously is elongating, it's extensing. So we'll see that these four characteristics, excitability, contractility, elasticity, and extensibility, are the four telltale characteristics of muscle tissue. And then finally, and something you're already aware of, is that there are three types of muscle tissue. There's skeletal muscle. That's what we will be dealing with this week primarily. There's cardiac muscle. We'll be dealing with that in far greater detail when we get to the cardiovascular system. And there's smooth muscle. Smooth muscle will be more highlighted in the digestive system as we look at the functions of smooth muscle in the digestive tract. Next week, you'll also be reminded of the histology of these three muscle types. So let's just take a look at the overall macroscopic appearance of muscle. Here we have a bone. You might recognize this already. This is the femur. And to the femur, first of all, I see a number of tissues that I want you to be thinking about. You've now come far enough along in your understanding of histology to appreciate that in front of you right now, there are a number of different tissues. And if you had small sections of these tissues under the microscope, you would now know what to expect to see. So let me start here. On the head of the femur, I see a white translucent tissue. You know that to be the articulating cartilage, or more specifically, the hyaline cartilage. I want you to think for a moment. If you were looking at hyaline cartilage, what would you see? Recall you would see the chondrocytes located in the lacunae. Here we have compact bone. Compact bone under the microscope would look like osteons with central canals and lamellae and canaliculi. I'm seeing tendon here. Remember, tendon is composed of dense, regular connective tissue. Under the microscope, dense, regular connective tissue, remember, has those long, stringy wavy patterns of tightly packed collagen fibers. I see here too some blood vessels, and recall that blood vessels are surrounded by a layer of simple squamous epithelia. So you can picture what that would look like, simple squamous epithelium. And all of your blood vessels are also surrounded by a layer of smooth muscle. And you've seen smooth muscle briefly previously, and you'll see it again next week. Finally, we see muscle, skeletal muscle. So skeletal muscle, recall, would be striated muscle with the very long muscle cells, the multinucleated cells. So just in this picture alone, I can see 5 different tissue types that you have been exposed to and should be able to imagine their appearance under the microscope. So muscle. You have hundreds of muscles in your body. We will name just a few dozen of them this semester. And what I want you to understand at the beginning is that muscles are attached to bones through connective tissue structures referred to as tendons. Tendons are typically a strong rope-like structure. They are nearly avascular, not completely avascular, but so nearly avascular that they appear as white. There's very, very little blood flow, which is why a tendon tear is so slow to repair. You will also see in the body another arrangement of this dense, regular connective tissue, and when it is found in sheets, it's called an aponeurosis. Now, in the pictures of the models that you'll be looking at, you're going to see some aponeuroses. There's 2 that come to mind. One, across the top of the skull, you'll see a white sheet of tendon-like material. Goes from the frontalis muscle to the occipitalis muscle. That's the epicranial aponeurosis. And then across the abdominal area, there is a flat sheet of tendon, the aponeurosis covering the rectus abdominis muscles. So here is that same bone and muscle combination, except now it has been pulled back just a little bit. And we're going to deal with the specifics of muscle tissue next week and again in lecture in a couple of weeks. But for now, I just want you to appreciate that muscle is composed of groups of muscle cells. So within this entire muscle that we're seeing here, there are actually groups, right, clusters of muscle cells, and each of those clusters or groups is referred to as a fascicle. So a fascicle is a bundle of muscle cells, and there's connective tissue in between the cells in these bundles. Each cell in a muscle is as long as the muscle is. So if the muscle is 12 inches long, then the cells within that muscle are also 12 inches long, and each muscle cell is also referred to as a fiber. So you'll hear me flip back and forth between calling it a muscle fiber and a muscle cell. They are one and the same. Now I want to be clear here too that the word fiber has multiple uses in anatomy. When we were discussing connective tissues, a fiber was a protein fiber, like a collagen fiber or an elastin fiber. Here, when discussing muscle, fiber refers to the entire muscle cell. And we'll see other uses of the word fiber when we get to the nervous system. At this point, there is a specialized organelle inside muscle cells referred to as myofibrils, and we'll see those again next week. A large part of what you'll be learning in this lab and in the next couple of labs, not only will you be learning names of muscles, but you'll also be learning approximately 20 different origin and insertion pairs. Each muscle in your body has a particular job in action. So you'll also be learning actions of muscles. And in order for a muscle to do its job, in order for a muscle to do its action, it must be attached to predictable places of the skeleton. Many of the bone markings that you're learning are in fact places where muscles attach to the skeleton. And when the muscle contracts, the bone is pulled in a particular direction. So these terms, origin and insertion, have very particular meanings. They are the two attachment spots for a muscle. The origin is defined as the attachment with less movement. Okay, so I'm just going to put less movement, and then I will describe one of these to you. So the origin is the place where the muscle attaches to the bone, where when the muscle contracts, there is less movement. The insertion is where there is more movement. By convention, the contraction of the muscle brings the more movable place, the insertion, toward the immovable place, the origin. So the insertion moves toward the origin spot. And you will be looking through your lab manual. There is a list of muscles that you are learning in today's lab, and some of those muscles on the list have an OI next to them. The ones that have an OI next to them, you must learn the origin and insertion for those, just those. And you will find that information scattered throughout the tables in your lab manual. You'll also be asked to learn the action of some muscles. Again, on that same list, you'll see some A's after some muscles, and for those muscles, and only for those muscles, you'll need to learn the action of that muscle. So let's talk about these bone markings for a moment. I'm showing you two examples here just to give you an idea of what you'll be looking at. On the left-hand side, this is the temporal bone. So this is the temporal bone. This is one of the bones of your skull. And there are a number of bone markings or bony features of the temporal bone that you'll need to learn. Number one, there's an opening on the outside. That's called the external auditory meatus. You'll also see it called the external acoustic meatus, and that is simply the opening in the bone for the ear canal, for sound waves to travel into your ear. There is a zygomatic process. Well, in bones, a process is something that sticks out, and this process is sticking out toward the zygomatic bone and will make contact with the zygomatic bone through that process. So that's the zygomatic process. There is the mandibular fossa. A fossa is an indented, smooth area. The mandibular fossa is where the mandible will articulate with the temporal bone and where the temporomandibular joint will be. But that's where the mandible will articulate with the temporal bone. Here there's a styloid process. A stylus, you know a stylus, a long, pointy type structure. Styloid process of the temporal bone. We will see there are ligaments and muscles that attach to that styloid process. There's another process, something that sticks out, called the mastoid process. There's a muscle that helps to control the flexing of your neck and the rotation of your head called the sternocleidomastoid. And we will see that that sternocleidomastoid attaches to the temporal bone in this place. So you get the idea that there are numerous landmarks or bony markings on many of the bones that you'll be learning. There is a long list of bone markings that you'll be learning this week, specifically for bones of the axial skeleton. Next week, in Lab 5B, you'll be looking at the bones of the appendicular skeleton. Another example of bone markings may not be where a muscle attaches, but here in the skull, you can see that there are sutures. So this sagittal suture is what separates one parietal bone from another parietal bone. The coronal suture separates the frontal bone from the parietal bone. There's a squamous suture. It's going to separate the parietal from the temporal lobes. And then there's a lambdoid suture, which is going to help separate the parietal bones from the occipital bone. So the sutures are a bone marking where the flat bones of the cranium come together. So you have quite a few bone markings to learn this week, and quite a few muscles and origin insertions and actions to learn this week. When discussing muscles, you'll see that there are muscles, for example, that move the arm or move the leg or flex the hip. And oftentimes muscles work in groups. They don't work all by themselves. They work in groups. And in those situations, the muscle that provides most of the movement is called the prime mover. The prime mover. Other muscles that assist that muscle are also sometimes called the synergists. Right? These are the synergist muscles. These are the muscles that assist in moving in some particular way. You know that synergy is when two things work together better than they could by themselves. So when muscles work together, there's oftentimes synergy involved. And muscles that oppose the prime mover and cause movement in the opposite direction are called antagonists. There are antagonistic groups. For example, the hamstring group in the posterior thigh is antagonistic to the quadriceps femoris group, the anterior group that extends the leg in the anterior thigh. Or you may know that the biceps brachii is antagonistic to the triceps brachii. Again, you'll be learning the actions of a number of muscles, just the muscles that are indicated on the 5A list. So muscles are named with various reasons. These reasons have to do with the appearance of the muscle, maybe its location in the body, what it does, its function, how it's oriented, and or unusual features that muscle does. So for example, as you're naming muscles, you need to pay attention to these words. These root words are going to tell you a lot about the location or the function of the muscles. So to start, the rectus abdominis. The rectus abdominis, rectus means straight, abdominis, in the abdominal region. So the muscles that go straight up and down in the abdominal region, your six-pack muscles, the rectus abdominis. Please, please, please make note of the last two letters of this word, IS. In the muscle vocabulary, you will need to spend a little bit of time focusing on the ends of muscles. Spelling is critical. And does the muscle end in IUS, EUS, US, OUS? There are lots of variations at the end of the spelling, and it's important that you spell these correctly. Other muscles are named for example, where they're found. The tibialis anterior is a muscle in front of, anterior to, the tibia, right? One of the long bones of the leg. Others are named for where they originate or where they insert. We're going to see the sternocleidomastoid. Sternocleidomastoid. And we're going to see that muscle goes from the sternum, right? Sternum to the mastoid. So that muscle is named primarily on the attachment places where that muscle attaches to the skeleton. One more feature about muscles is that those fascicles, those groups of muscle cells that I referred to previously, have four basic patterns of arrangement. You will see some circular muscles in this lab. Okay, circular muscles. And I just found a typo. So this is not spelled correctly. This should be orbicularis. So refer back to yours. Correct. The spelling is correct in the handout. But the orbicularis oris, and the orbicularis oris is the circular muscle that goes around the eye. There are a number of sphincter muscles in your body, circular muscle groups, that are also found in your digestive system. There are muscles that are considered convergent. That is, they look more triangular. A good example of this is the pectoralis major muscle of the chest. You'll see in the diagrams that it has basically a triangular shape. There are muscles referred to as parallel muscles. They are long muscles. Your biceps brachii, for example, is a parallel muscle. And one characteristic of those long muscles, those long parallel muscles, is that they have a central body, or what's called the belly, or the gaster of that muscle. So when the biceps brachii contracts, the body or the belly or the gaster of that muscle is what pops up. And then pennate refers to a feather-shaped muscle. And these are some of the strongest muscles of the body because the fibers are arranged in an oblique pattern that gives them a feather-like appearance. And you'll see all of these fascicle arrangements in different muscles that you'll be learning in the next couple of weeks. Again, how are muscles named? Again, be watching for those clues. So, is the muscle named for the shape, like the trapezoid shaped trapezius, right? The trapezius. If there is a magnus, right? Then there's probably, that's the largest of something. We'll see an adductor magnus. There are, for example, the pectoralis major. If there's a major, there's also probably a minor, and there is a pectoralis major and a pectoralis minor. You're going to see the gluteus maximus and the gluteus medius. There is also a gluteus minimus that we will not be. You will see muscles named for the number of heads or tendons. So the biceps brachii has 2 tendons. The triceps has 3. Some muscles have the word flexor in them because that's what they do. They flex. The muscle, when it contracts, causes the skeleton to flex. Some muscles will be named because of where their location is. There are some muscles that have internus in their name. Others are named after their length. Very long muscles maybe have the word longus in their name, and very short muscles would have brevis, brevis in their name. So again, be watching for and focusing on these prefixes and suffixes and root words. They will help you in naming and understanding the muscles. So your job today is to learn the bone markings of the axial skeleton. You've already got your list and organized that. You've already found those bone markings in your lab manual and in the lab atlas. You need to start applying those bone markings, especially to the muscles that you need to learn the origins and insertions for. You'll also be naming a number of muscles, essentially the muscles that are found associated with the axial skeleton. So you're looking at the muscles of the face, skull, thorax, abdominal region. And you have your lab manual, you have your lab atlas, you have your textbook, and don't forget you have Mastering and PAL to use as ways of looking up and confirming these muscles and bone markings. You also have your skeleton. Last week you used it to name bones. This week your lab instructor will be giving you some ideas to use the felt and some of the other stickers to label your skeleton. Be listening for specific instructions from your lab instructor.